Thanks so much, Lance. I'm happy to round out this really intellectually stimulating conversation here. All right, so as Lance said, I'm a program manager at DARPA. And first of all, and thank you for the organizers. This is my first APS meeting, and it has been truly wonderful. Today I'm going to share with you what DARPA is really doing. We heard about a lot of sci-fi things. And I wanted to, before I do that, I want to talk about, it's really interesting how people end up where they're at. So I'm actually a cognitive neuroscientist. Before I joined the government 15 years ago, I worked at Johns Hopkins University in the Department of Otolaryngology, doing some path integration studies and behavioral studies. Went over to the intelligence community, worked in what's known as GeoInt, FBI, other three-letter agencies, and ended up at DARPA about 2 years ago. And I believe one of the previous speakers spoke about Tristan. Tristan was the brainchild, I guess you could say, behind Biostasis. But the nice thing with DARPA, they keep fresh blood in, right? We have expiration date, pun intended. We all have an expiration date there, and so typically the sweet spot is 3, 4, maybe 5 years. So I came in and I inherited Biostasis. And so it's been really fun getting to learn the bio and pretend to be a biologist. Next, please. Actually, I control this. All right. Yeah, we're very spoiled at DARPA. We're used to people running things for us. All right. So, okay, before I actually launch into the genesis of Biostasis, I thought it'd be nice to tell people what is DARPA or what do we do? And so those of you who are unfamiliar with DARPA, it actually was initially founded as the ARPA, so Advanced Research Projects Agency, in 1958, and this was in response to the Sputnik launch, right? And the goal here was to never be under that sort of strategic surprise ever again. But we also wanted to spearhead our R&D, our research and development agency that was really pushing the U.S. to be on the bleeding edge of technology. And so to do this, I say DARPA has a really unique formula. We really embrace high-risk, high-reward projects to support the DoD, the Department of Defense. And I think that formula deals with the people, the culture, as well as the processes. So DARPA program managers like myself, or like Tristan and some of my other colleagues, are really responsible for conceiving and managing the programs through its life cycle. But our programs are kind of different from like NIH or NSF grants. They're really metrics-based and bounded to ensure success. And then lastly, I think the sort of secret sauce is that also DARPA is not afraid to fail, right? We feel confident that an idea that is kind of guaranteed to work is kind of not where we should be placing our bets. And then I think the results of these are pretty evident. The outcome of this formula has been pretty successful. DARPA has been an early investor in technologies that really fundamentally changed our world, right? And changing our lives now. So things like, drones, stealth fighters, ARPANET, what's known as the modern internet. I believe you guys have Vince speaking a little bit later, or maybe tomorrow. GPS, that's a DARPA outcome. And then lastly, those of you who may be familiar with the Global Hawk, which is really a RPA, remotely piloted autonomous sort of ISR capability. All right, and so where I sit, we have several different what's called TOs, or technical offices, and I sit in BTO. And actually, BTO is actually the baby of the agency. It just stood up about 10 years ago or so. And our office is really looking at, obviously, biotechnology. And it's split into 2 areas you see here, warfighting readiness and operational biotechnology. And then that warfighter readiness is further broken down by operational readiness and warfighter health and recovery. So these will include things like MCMs, medical countermeasures, diagnostic assessments, and tactical care. So a couple examples, we just launched a program, a fellow program manager has a program called GoldieVac that looks at like complex polytrauma. And then we also have DTC, the DARPA Triage Challenge, that's really looking at how do we deal with MCIs, mass casualty incidents. Sorry, DARPA loves its acronyms. I'm spelling everything out for you guys. And then under the operational bio, there's that's again further broken out with sustainable operations and operational advantages. And you could think about things about resilient infrastructure, sensors, novel methods and materials, but also how do we build resiliency into our bioeconomy chain. So we have a really fun program called Cornucopia. That one is really looking at deployable on-demand production of really appetizing microbial origin food. So how can we create food from air, water, and electricity with minimal to no supplementation. And then today, obviously, I'll be talking about biostasis. All right, so as you heard, Biostasis was launched back in 2018, 2019, and we're actually wrapping up our final and fifth year. So again, we don't have these long programs that go on, these legacy programs. They have, discrete beginning and end dates. And so as part of the Warfighter Health and Recovery, it really aimed to address a really urgent problem with battlefield injury care. So before we delve into how DARPA approached the problem, I want to discuss, what was the state of the art at the time, or currently is still this kind of state of the art for treating battlefield injuries to illustrate really why this program is so important. So the main issue the military faces, or DoD, is that the treatment of our injuries and our boots on the ground is really too slow. It's really reactive, and it's also pretty much dependent on cold chain. And when you think about the future battlefields and fights, right, we need to be able to increase our operational flexibility. So traumatic injuries on the battlefield can, quickly turn fatal. So with over 50% of our combat deaths really occurring within that first hour. And you heard about the term, I believe Jane threw out the term of golden hour, and that's a really critical window where outcomes are greatly or significantly improved if proper medical care is provided quickly. But when you think about, again, in the future, we may not have a whole hour, right? You may need to extend it to maybe days or even weeks. So logistically right now, not just treating our injured service members, men and women, but think about the maintaining the actual countermeasures. Things like reagents have to be refrigerated or frozen, which really imposes a significant cold chain burden and really limits our availability and lifespan. And then lastly, infections are another huge challenge that we deal with, especially in medical facilities. It's really limited, so the state of art treatment really depends on identifying the infection and then providing the appropriate countermeasure, which is often not possible in the field. When you think about battlefield care, people need immediate care. All right, so knowing these issues, DARPA stood up the Biostasis program to extend the golden hour, again, to multiple hours or even days. And we really envisioned meeting this emerging need by slowing down biological time. So you heard about kind of instantiating that biological pause button and allowing the situation to sort of stabilize. Importantly, though, not just inducing this biostasis state, we wanted it to be controllably reversible, so that the injured person on the battlefield can be evacuated to more advanced medical facilities or where biology can be resumed and treated. But if you think about it, I think you heard today about all the different applications space. A biological pause button is not important just in acute trauma care. We think that it could prolong the lifespan for like really fragile biological reagents, whether that's like antibodies or even blood. Some of our DoD partners are pretty much interested in like shelf-stable blood. So the benefits really are hugely beyond the DoD needs. And I think a little bit later, if I have time, I'll talk about some of the civilian applications as well. All right. I think some of the previous speakers hit on this point, so I won't belabor this too much. But is pausing biology even possible, or does it seem like sci-fi? And I think the speakers did a great job to show that there's actually many examples found out in nature that gives us clues how biostasis can be achieved and adapted in humans. And a lot of our in the early stages in phase 1, a lot of the research organizations that funded took a look at things like cryptobiotic organisms like the tardigrade, the cute little water bear you see there, and toads. And basically, they want to look at how these organisms really enter these low metabolic states, such as hibernation or torpor. And I really want to highlight that water bear, because it just blew my mind. Again, not being a biologist and working with some of the universities, to see it really survive the extreme depths of vacuum of space, freezing temperatures, and being completely desiccated really blew my mind. So we wanted to consider, how can we mimic some of these organisms and this biostasis states in humans? All right. So again, today, I know I got warned about the time, and I'm trying to speak really quickly, but I really have some really exciting results from 3 groups that we funded, that the Department of Defense funded. And so you see here we had Wyss Institute developed really small molecules that stabilize proteins and really interact with the hibernation pathways. And that picture on the top The left is just depicting these sort of what they call molecular chaperones that work universally by locking the enzymes into these like inert conformations. The second group that received DARPA funding was the University of Colorado Boulder, and they were taking an approach to change the entire biochemistry within the cells, right? So they were forming these cross-link gel-like environments that pretty much restricted reactions from occurring. So in that picture, the hydrogel was causing what's called like intracellular crowding and preventing that molecular diffusion. And the last group that unfortunately I don't have time to talk about today was the University of Wyoming, and they were the ones that were adapting those tardigrades, sort of like proteins, these IDPs, to work with human cells. And just to reiterate, these three approaches really should be able to pause the affected cells from undergoing further infection or damage, whether it's from injury or through natural degradation. And importantly, these are all athermal. So you may have heard of some things that are temperature dependent. All of these DARPA programs was athermal, so that's really cool, meaning they didn't need cold or freezing temperatures, right? Another important point here, DARPA does like its phases. So this was really structured in 4 phases, which I thought was really important, from increasing the complexity from going in, simple cells and dishes all the way working their way up to really complex animal models, like in pigs. And so we're in the final stages where some of these performers are demonstrating some of the validity of their approaches in these larger animal models. All right, so next, here goes the exciting part. So I want to show you some of the results that we had coming out of the program. So again, recall that first approach, the small molecule or molecular approach. These actually identified several candidates by designing really small compounds using kind of like simulations, molecular simulations, to stabilize the biomolecules inside the cells. And again, this kind of limits their ability to interact and compromises that biological time. And so one of the lead compound that was tested in these lung chips, right? So these chips are designed to resemble the human lung and to evaluate how they protect against like irradiation, right? So looking at the bar here, you can see the bars represent the cellular survival in these chips. And as you can see from the red arrow, by adding the biostasis compound, it actually improves survival over 5-fold. And in this case, they simulated how this compound interacts with DNA, which is shown in the blue, and the black molecule is the compound, and it's theorized that physically fits into the groove of the DNA and stabilizes the damage incurred by the radiation. So pretty exciting stuff there. Moving on from the cultured cell experiments, they moved on to actual living organisms, and we looked at some of these biostasis compounds in tadpoles. And actually, tadpoles are really, really useful models because their behavior is really easy to observe because they're transparent, right? So you can do this live imaging, and you can see processes like their actual heart rate, right? So here, the biostasis compound was administered to the tadpoles, and as you can see in the graph on the left, that their movement quickly drops to almost no movement as compared to the untreated group in blue. And that happens fairly quickly, within 2 hours. But recall that one of the aims of the program is that we wanted the biostasis to be reversible, right, controllable. So what you can see there is after 2 hours, the movement begins to recover and actually goes back to normal within 24 hours. I'm going to see if this video actually works. So on the right there, that's with our the biostasis compound added. And pretty quickly, you could see that the slow down in movement. It's pretty evident. And because I'm kind of cramped for time here, I'm not going to let it run, but importantly, if you trust me, they actually return back to normal movement. All right, so here's some really fun other results that we had with this program. So we aim to develop the biostasis compounds that broadly slowed biology, and we saw that affect everything from cells on a chip, the lung chips, to the tadpoles I just showed you, but we even see it on food. I think this has a lot of implications when you think about like food security, right? And here you can see the spoilage of bananas at high heat. So it's like 300 degrees Fahrenheit, and it's showing the biostasis-treated bananas are actually more resistant to this enzymatic heat-based degradation or browning, right, compared to the untreated bananas. And this is at multiple time points. So, and the more interesting thing is below there is that room temperature. You see that it's even more stark with the biostasis treated bananas lasting for 9 days with minimal degradation compared to the untreated ones. So I think we're really looking forward to see how these compounds could actually preserve biomedical supplies too. But it really opens up really intriguing possibilities for a really broad spectrum biological preservation. And then another application that isn't really in advanced testing is the possibility of using these biostasis compounds for tissue preservation, right? So whether that's following the battlefield injuries or things like dismemberment or for organs during transplantation. And so this graphic here just shows that most organ donors, for most of them, only a small fraction in orange are actually able to be used. And the reason really includes that these organs degrade during that wait. You have that logistical challenges, right? So this really highlights the need for improving organ preservation to deliver on those really life-saving potentials, and really has immense benefits outside of the military as well as civilian populations, right? And because I'm cramped for time, I'm going to skip that. And then one of the latest trials is with the donor hearts. This really, we just had our PI meeting back in January, so this is really sort of hot off the presses results that we saw with the 3 biostasis candidate molecules that were actually really effective in reducing that oxygen consumption. You can see on the right, 40% reduction. And so why is this important? Reduced oxygen consumption really indicates a slower metabolism and a lower energetic demand. That's exactly what you want from organs that got to survive outside of the body, right? The nutrients are not available. So in this experiment, the donor hearts were perfused with a biostasis candidate, and I believe it was about in a perfusion chamber for about 6 hours. But immediately after, what's important is that immediately after they washed out the treatment, the heart returned to normal levels. So that's kind of confirming, again, that important point that the biostasis is indeed reversible. And as I mentioned, not only is the metabolic demand reduced, it really has important ramifications for organ health and quality. So these graphs from the researchers here examine the expressions of like markers for cell death, things like hypoxia or inflammation. And as you can see that the hearts that underwent the biostasis actually showed lower inflammation, hypoxia, as well as cell death. So they're pretty much in better shape during this transplant process. Right, so the results all from these small molecule biostasis compounds are pretty intriguing, but I also real quickly would like to show you some results that came from our intracellular polymer group. All right. So the thinking real quickly behind this is that this approach is that the biological activities really depends on free movement of molecules to participate in reactions. So by crowding the molecules, you're really limiting their free movements, which decreases the reaction rates as the molecules get hindered, right? And so this is seen in bacteria in 2 metabolic states. In a metabolically active state, the single molecule tracks show they're free to diffuse really long distances. And then in the metabolic inactive states, these molecules are really locked into these inert regions. And so knowing this, one of our biostasis groups designed a strategy to really replicate this crowding. And I'm going to show you that right there. The overall strategy, again, was to introduce these monomeric compounds inside the cell. And when they're inactive, they freely diffuse. But however, they can obviously polymerize to create this cross-link hydrogel. And so when the nice thing about this, again, it's that it's inducible, reversible, and it's achieved inside of the cell, right? So there's no observed detrimental effect on the normal selectivity when the biostasis is lifted. So a lot of exciting results. Another area, I don't think that was quite conceived in the very beginning stages, but came up with one of our partners, is that we want to be able to test this system in burn wounds, and the results have been quite promising here. I notice I'm running out of time, but basically, if you look at that cartoon on the top right, it depicts a burn that's bandaged with this biostasis agent. And the solution is absorbed by the cells in the wound. And as you see in the line graph there, how well the wound is healing, it's really controlling that damage. And so as you can imagine, the biostasis is kind of giving this kind of protective treatment for the healing process. All right, so lastly, I'm not sure if you guys have been tracking the stand-up of ARPA-H. So that's the Advanced Research Agency for Health. And they're really looking at how can we take some of the technologies that DARPA built and funded and now apply it to civilian applications. And so a lot of the approaches that we pursued, as you can think about, has other implications for, not just extending the golden hour for our DoD boots on the ground, but also for patients that may be far removed from medical care. Or, as some of our previous speakers spoke about, maybe even long-term space travel. And so we're really looking now out to the future for transition partners to really help meet the regulatory and drug discovery guidelines so we can move these forward. And I think lastly, I want to end on there's opportunities. I know there's a lot of folks here from academia. DARPA is really, again, charged with doing these breakthrough paradigm-shifting solutions, and we're always looking for great partners and agencies and funding opportunities. So if you'd like to hear more, just come chat with me during the conference, and I could talk to how we could bring your bright ideas to realization. All right, I'll end it there. Thank you.
Thank you, Dr.
Symposia in future years. So I thank all of these speakers. I'm going to start with asking the panel if they have questions for one another. Not technical, but things they think that the audience might want to hear but doesn't know enough to ask the right question. So, to the panel. Yes.
I have a question for all those guys. So it seems like there's an underlying assumption that when you have biostasis, when you get the stasis, that then it's okay. So you take functions out of the body, you slow things down, and then it's going to come back to normal. So what do we know about that? And what if we're wrong? I mean, what if something interesting is happening during that biostatic time or that extracorporeal time? Are we studying it and asking, is it exactly the same, or is something interesting that we might learn from actually happening during that period? That's for all of you.
Sam, okay.
I'll jump in first. So, I mean, certainly from our perspective, all the work that we've done in the lab, we've focused on normal functional outcomes that we don't want. I mean, the worst thing that could happen with resuscitation is you get somebody back that's alive but not really alive neurologically. So as best we can tell, we're shooting for a normal, healthy person after we do what we do. So that's our goal, at least.
Is it the same normal, healthy person, or,
Yeah.
Are we the same tomorrow as we worked out today?
That's a really deep question.
Maybe they're
Right.
better. I don't know.
So I'm only going to give one of these people a chance to answer, because otherwise we'll only get a few questions in. But Lance, why don't you speak to that too? Because I saw you nodding.
Well, I think it does have to do with kind of what is our view of life. I mean, what are we, and am I going to be the same tomorrow as I was today, or am I the same today as I was yesterday? And I think those are difficult questions to understand. I think in the gross look, when we study people that have gone through the closest things that we know to being temporarily dead for an extended period of time and then are resuscitated, what we know is the vast majority of those people, if they recover function, they mostly feel like they were quite similar to the way they were before. But I will say, having talked to thousands of survivors, they also say they view life a little bit differently. They many times are much more appreciative of little things that maybe they didn't appreciate before, and they may have some small deficits that I don't recognize, but they, recognize. In Philadelphia, this is very public, so I'm not, I resuscitated Lawrence Saltzman, who's a Philadelphia photographer. And he was the very second person who was cooled down in the ICU 2 miles from here. And he made a fabulous recovery when everyone really assumed he would never recover any function. And in fact, they had called his wife in to tell her that he was brain dead and that they would be discontinuing care on Lawrence. And she literally showed up on my porch because she knew that we had just started doing cooling at this point. And Lawrence made a fabulous recovery, and I knew things were going to go well when his wife came to me and said, well, he was able to say some words to me this morning. I was like, oh my God, that's fabulous, because he had been comatose for about 3 days, and the neurologist had said he would not recover. And she said, well, he spoke 5 of his 7 languages. And I said, well, you know what? And she said, well, I don't speak the other 2, so I couldn't test the other 2. So Lawrence made a fabulous recovery, and he's spoken about it, and he is alive today, and I get cards from him all the time. It's part of what keeps me going, you know. And so, is he the same person? You could ask him.
Bonnie, all right.
Yeah, I would make the point that that state of normalcy, it varies, and it certainly takes a great deal of time for some individuals. And what they are willing to go through and what they're willing to put up with is, I think, what's critical to what makes their life meaningful or not. So say if that were his 2 favorite languages that he could no longer speak, that he might feel, or she might feel differently. Or if brain comes back but maybe kidneys do not, and that means a life of dialysis. So that normalcy, it's going to vary, and oftentimes when patients will ask me, well, what should I expect? Oftentimes I don't know for sure, and that it's a day-by-day process, which, we have to talk about, and I let them know that honestly.
I think it's an interesting question. I think for DARPA, like I said, we started, it was very much, the program was structured like a drug discovery program, right? So like I said, from simple to more complex models. So the data that I have is only just on animals. And we know we can look at behaviorally, it seems like they're returned to normal, but we can't interrogate their affective or cognitive states. But moving to that human, so doing these preclinical trials to move to look at some more of those human style trials are really interesting to me. And I know I was kind of rushing, but at the end we're handing off these technologies to ARPA-H so that they can kind of move it into that phase 1 clinical study. So it's kind of like, we don't know, more to come. Yeah.
I think it's both a societal, ethical question and an individual question, right? The question of when do I want further intervention is very individual, and it's something as we age, we become conscious we need to make these decisions at various points for ourselves or our loved ones. It's not just a societal decision. And new medical technology, medicine has always advanced by trying things that look possible, and not always with good outcomes. So getting from a few percent, 0 chance to high percentage recovery to good life is a process, not a one either or. I'm going to turn to the audience for questions. I'm going to ask the panel to try and refrain from all trying to answer every question because we'll have so few questions if we do. So I want you to direct your question to a panelist if you can. And I'm going to go to one of our planners first, Sam.
Yep. Ron Fairman.
Ron, this is Sam, that's Ron.
Ron
Sorry.
Fairman. So I think Jane brought up a really interesting question, and I'd like to ask Bonnie a question about it. So it's pretty well known that the length of time you're under hypothermic circulatory arrest impacts on the ultimate recovery. And one of the things that we don't know very much about is cognitive impairment, following, quote, successful aortic reconstruction, valve reconstruction, et cetera. Can you comment about, have there been any new learnings that, because cognitive impairment to the people that have it is profoundly disabling.
Right. You know, and that got to a point that Nina and I were talking about, the prefrontal cortex. And I think the bottom line is that the longer we go on either cardiopulmonary bypass or circulatory arrest, that very subtle cognitive impairment, we know for sure that that happens. I mean, we have a lot of literature that supports that. And it can be very finite, a very specific area, whether it's language, spatial processing, or whatever it might be. You know, I mentioned an individual highly functioning who had mentioned to us that he could no longer do the New York Times puzzle any longer, or it took him an inordinate amount of time. Now, that was ultimately not that important to his quality of life, but I mean, certainly we need to try to keep our time on that suspended lifespan, whether it's on bypass or circulatory arrest. We need to keep that as short as possible, because yes, we know with very specific neurologic testing that those deficits really can be picked up. So it's not just stroke, it's not just you know, piece of plaque that went to an area of the brain, that you can see it on a CAT scan or an MRI, but instead it's that neuropsych testing that really picks up the deficit.
Okay. Question over here.
Hi. I'm Alan Spradling from the Carnegie Institution and the Howard Hughes Medical Institute. I want to speak to Benjamin Franklin's observation that is the inspiration we hear for this symposium. So he saw flies that he poured out from a bottle of Madeira, wake up in a few minutes, and then a couple of them flew away. Now, I don't think maybe everyone realizes, I've worked on fruit flies and flies for more than 40 years, okay? And they're very attracted to wine when it's been opened, by the way. And I wonder, what is the history of anyone ever trying to replicate his experiment? And I can tell you, to my knowledge, and there's thousands of really good scientists who work on fruit flies, and no one has succeeded in making flies come back after 3 months of submersion in wine or anything else. So I just wonder, which is more likely, do you think, as the explanation for his observations? Biostasis for 3 months, or the flies flew in there when he wasn't looking and then he dumped them out on his glass?
I think we all have that question in our heads. But
So can I just, so Alan, I tried to find that somebody had replicated. I looked up and I found 3 places where people said, I tried to reproduce that and I couldn't get it. And then one said, I think the flies were drowned and came back, but what does it mean to be drowned rather than just sitting on the edge of the bottle and being...
Yeah.
or they were drunk. Question up above.
Hi, Richard Linsky, Michigan State University, and the question for Dr. Becker. I found the replacement of the mitochondria to be a fascinating aspect of your work, and I was wondering if you could elaborate a little bit on how you do that and how you show that the mitochondria have actually been replaced.
Well, so not my idea, and when my young people came to me, I said, it's the craziest thing I've ever heard. I wouldn't even hardly let them do it. But people should know there are 2 human studies going on right now in the United States on mitochondrial transplantation. One in Boston, one in Seattle. They are ongoing, and they look very, very interesting. And the way it's being done right now is they're using tissue from the individual And there is a rapid technique where you simply take a very small snip of tissue, and you can actually isolate mitochondria in 20 minutes at the bedside if you use a quick technique. Now, in the laboratory, I should say, I am a mitochondrial biologist, and the way we would do it takes an hour, hour and a half, but this is a bedside technique that was developed by the investigators at Boston Children's Hospital by Jim McCulley. Please look up his work. He's published on this. And you get highly viable mitochondria in 20 minutes, and those are then being injected either into the organ of interest or into the IV system. There are so many questions on this right now. It really has people quite, I'll say, abuzz, all right? Because no one is exactly sure kind of what's going on. The evidence for uptake is kind of sketchy right now. It is really clear that it does something. Whether or not they actually get to the target organ is completely unknown. There's some weak evidence for that. It is clear that it has a profound effect on the biology, though in all the systems that have studied it, they, like you see things. We have a lot of data that I can't really talk about right now on this because it is what all of my young people want to, like everyone just wants to stop doing what they're doing and work on this. And to be honest, like I want mitochondria to be infused into me as soon as possible, okay, because my brain needs it like this morning, do you know what I'm saying? And so I do think we're sort of moving in that direction also.
And we do one right here at the front, and then there was one at the back.
Hi, I'm Samuel Russia, University College London. First of all, thank you for such an interesting seminar. My question is for Dr. Becker. Please correct me if I misunderstood this, but it seems that giving oxygen after a long period causes cell death. But why doesn't it cause cell death if you give it very early at the beginning of death?
So you absolutely heard what I was trying to provoke people to really think about. And it's still a bit of a mystery, but we do know that during the period of time without oxygen, I'm going to use a simple analogy, your body is not stupid, and it begins to scavenge molecules from lots of places. And part of what it takes apart as it's without oxygen, so it doesn't have a normal substrate, it begins to scavenge a number of the antioxidant defenses, okay? They become nutritional. So do some of the structural proteins, okay? So do some of the lipids. And so what you essentially have, if you will, is you imagine a house that's had a hurricane or something like this, all right? And you're without any kind of energy, and you've now weakened a number of the structures, okay? And now you sort of go back into the house after a period of time, you smell gas. You say, maybe this is not the time to turn the furnace on, right? So maybe what we need to do is kind of gussy things up, fix them up, clear the gas out, and now I can restart that furnace safely. Now, if you didn't have a nose, right, you're just unaware of what that is. And there's very good evidence that, in fact, in a way, very directly, it is almost as if our cells are combustible. And remember, we respire. We derive energy in the same way that things are combusted. And so what we think happens is with that prolonged period of being without oxygen, the cells are sitting there, they're quiescent, they're not dead, but they are vulnerable, okay, to starting metabolism in a robust, like full metabolism. And so that's where maybe sort of this little bypass into biostasis for a bit, allowing cells to reassemble the proper mitochondrial electron chains, the way we handle all of these very hot molecules. energetically hot molecules that we're handling, okay? Like, you're doing it right now with no problem, okay? You're not blowing up, okay? And so you do it very well, but if I deprive you of oxygen for a period of time, your cells will no longer be able to do that without what I think we could reestablish with a period of stasis.
One in the back.
I'm Ellen Winter from Cambridge, Mass., and I have a question for you, Dr. Becker, about mitochondria. Mitochondria dysfunction has been supposedly part of long COVID. And I'm
Yes.
wondering if anybody has considered injecting fresh mitochondria in long COVID patients.
It's exactly, so the person who described much of that is Doug Wallace, who's here in Philadelphia, and I shared a laboratory with Dr. Wallace for 10 years in the mitochondrial center at CHOP. And what he described is just fascinating, that there appears to be a COVID molecule that, so mitochondrial DNA is a big circle. It's like bacteria. It's not like our genes. And there appears to be a protein that sticks on, and it actually prevents the reading of that, and it just stops at a certain point. And he has described that and wrote about that about 2 years ago. It's a new mechanism of heart failure, and he found it in the people who died from COVID. And so, yes, it's exactly what I want to do. I've proposed it in an NIH grant that they trashed, okay? So yes, the answer is yes, and we ought to do it, and I'm ready to try it. If I had long COVID, boy, I'd be thinking about it. And I might add, you mentioned the children, that there are groups of children that have very profound mitochondrial deficits that are just terrible, okay? And these children are in very bad condition. And you can imagine having a child who at a very young age is having repeated horrible hospitalizations and barely able to survive. And you can imagine that those families are very, very interested in this as a therapy. And there are now, I will tell you that with this advent of mitochondrial transplantation, there are at least 10 startup companies that have already emerged around the world that are looking for realistic ways of using the technology that we have right now to try to ameliorate some of those children. And I predict there are trials that will be happening very soon.
If we can have some questions for people other than Lance. Just because it's a possibility. Upstairs in the middle row.
Thank you. Mike Out, Brooklyn, New York. I have a question for Dr. Meyenshine, or anybody else who wants to jump in and try and answer it. What does the prospect of resuscitation or resurrection or biostasis do to organ donation? And the patients who are waiting for an organ. We know that therapy works, and yet presumably the incentive to donate one's organs will be diminished by these prospects.
That's a great question. Others may have different answers, but it's a great question that has been discussed a little bit by some. And the literature is and the understanding about what causes people to be organ donors or not is so poor. I mean, we don't really know what causes people to be willing or not. I mean, there are various studies in various places, but frankly, it's really different in Arizona than it is in New York, apparently. And so it's hard to predict what the impact might be from what I've read. Maybe somebody has local experience that would be relevant.
And of course, there's also the counter influence that, as we heard, some of these technologies improve the survivability of organs out of the body to be able to transplant them. So.
Right. Yeah, I guess I'm optimistic. So I always want to see the glass half full. I would only hope that it would increase organ donors, right? In that knowing that your organs are going to be maintained for much longer. So that's exactly what they're looking at ARPA-H. So initially, like I said, Colorado Boulder V's have been looking at functional organ transplant, and now that we're handing this over, right, DARPA de-risked all the hard science, now we're handing it over to another government agency. That's exactly what they'll start looking at, right? They'll do some more PIC studies and hopefully get to clinical trials, but can we sort of maintain these transplants for longer? And then the other behavioral scientist in me also wants to see, like, how does that change public behavior and public perceptions? So I think that's a fascinating question.
This question is for anybody but Lance Becker. I take these instructions seriously. My question is whether studies of human sleep provide any kind of information that's useful in this work on biostasis.
Seems like nobody. But I'm going to ask Bonnie
Probably.
because there's also anesthesia, which is another kind of biostasis, if you like.
Right.
So,
Although I would comment that when someone is under general anesthesia, I know a lot of times we as practitioners say you're going off to sleep. It is not a sleep state, and that is pretty clear. I'm not sure that sleep studies would give you the same information in terms of biostasis because you're still live and functional. There hasn't been any type of injury that you're trying to protect against or trying to resurrect cells that are in the process of potentially dying. That would be my only comment.
It's a great question that probably connects with studies of hibernation. So there are a lot of studies now about different types of hibernation and what they mean, and is it a kind of biostasis or is it not? Because it's not really suspended all of animation, but it's suspending some things. And I know some people have said, well, how does hibernation in bears compare to sleep in humans? There have been some people discussing that, but I don't know that they found anything helpful yet.
I just had a quick comment about hibernators hibernate to get through the winter because they can't get food, and it's not necessarily a state that helps them with a cardiac arrest or shock. I mean, certainly the basic science might be helpful to us, but the hibernating state is not necessarily one that will protect them if they have a cardiac arrest.
Go to Nina in the front row.
Thank you. The panelists have addressed lots of wonderful therapies that draw on tremendous numbers of people, a lot of money, generally focused on single individuals. What I would like to know is, where do you see some of these technologies being translated to sort of more normal life circumstances? Joanna, you spoke to slowing the deterioration of bananas, but presumably some of these molecules could be used, that are being developed, could be used in other applications. Hypothermia, we see all of these people doing DIY plunges to try to slow their metabolism and prolong their life. I mean, these are sort of household hacks, but where do you see some of the legitimate technologies being used in more real life, general, out of hospital settings?
I mean, myself, I'm really interested in the biostasis compounds or mitochondrial transfer. I would love to see something that is developed that could be used maybe in a mass casualty situation. We have a lot of gun violence going on currently. Like, is there anything that you could inject or give on the scene that would allow that individual to survive until they can get to an emergency room or a hospital?
Right.
I think that would be exciting.
Yeah, I think I talked about a little bit about future applications. So we talk a lot about the golden hour, which is really going to be a misnomer when you're talking about these long combat operations, where, like I said, it's going to be not hours that you're trying to buy. The U.S. may not have dominance in terms of the airspace, naval, maritime, and so you're trying to keep people at hold for days, weeks, unfortunately maybe months. So maybe looking at more longer timescales. I think will be interesting. And then coming in, it's always interesting when you come in and you inherit a program and you start looking at it from different angles. And I was thinking about the hydrogels that some of the researchers developed, and they have made great, got to great diffusion depth, so they got down into the skin. But wacky idea again. Like, what about using it as a prophylactic, right? So it's inducible. You don't have an injury, but you have it sitting there. If you know someone's exposed to like maybe some kind of radiation or exposed, how can you use it as a prophylactic?
Question in the middle down here. Wait for a mic.
Thank you. I'm Joel Cohen from Rockefeller University, New York City. It's wonderful to have this high technology to cure people or to try to cure people who have been injured. Are we adequately investing similar intellectual and financial resources in prevention? Specifically, how many of the injuries from gunshot wounds could be prevented by high-tech research, social science research, experimental public health research? How much injury to our young men and women in the armed forces could be prevented by exploring alternatives to violence? Why are we not paying attention to prevention?
Anybody on the panel want to take that one on?
We all agree.
Yeah.
We agree.
Yeah.
We're talking about a very narrow slice of what goes on in the world today, right? We're talking about extreme situations and what we can and cannot do in them. But at the same time, there are a lot of other problems in the world, and how we invest our limited resources is one of those things that doesn't come up in this discussion, but certainly we all need to think about. Back there, yes.
Rich Schifrin, Indiana University. Dr. Arthur talked about molecules that would extend the period at which recovery is possible. And Dr. Becker talked about a cocktail that would allow a brain to recover function when oxygen would kill the brain. I understood it probably because it reversed the mitochondrial death switch, perhaps. I'm wondering if we're talking about the same kind of molecules with the same function, or are we talking about two different processes that are operating by totally different mechanisms?
Yeah. I probably let you take it, but
Well,
I was
really quick, I think it's different
different,
mechanisms.
yeah. Mhm.
But please.
Yeah, but I think you're also getting, I'm going to try to pull the strand too. I think you're getting at another interesting point. Again, we don't have enough time to bear out, to look at all these different hypotheses. But one of the other things I thought about in inheriting this program is conceivably, all the experiments were just looking at these one pathways, but what if we do kind of like a polytherapy, right? So some of the treatments that some of the other panelists talked about, can we use some of these compounds together and you kind of see, instead of this additive effect, the synergistic effect. And we saw some of these in some of the earlier studies. I'm not blank, I can't remember if it was Feast, it may have been Wyoming, that looked at state-of-the-art like trehalose, a disaccharide that's used to like maintain certain foods or makeups, and can you combine that for maintaining blood? And what they unfortunately did not find that it wasn't a whole blood stabilization. They only stabilized some components. But that is like, again, the next step, right? The next frontier is seeing if you could combine some of these therapies.
Question way in the back.
Jonathan Lear.
The mic is on.
We're not hearing.
Psychological studies about people who have come back. The dead, not just cognitive function, but how they felt about it, what their emotional lives were like, and are there any programs of psychological treatment and therapy for people who've come back from these experiences?
And yes, there's been some work on this, and it's quite variable as how people recover from a cardiac arrest or actually any kind of critical illness. But it's tough to treat it. I don't know if Dr. Becker can add more to that, but I was going to comment earlier too that just being critically ill from whatever causes you to be critically ill, whether it's a cardiac arrest or trauma or sepsis or lung failure, we've recognized what's called the post-intensive care syndrome. And we get the patient out of the hospital and they go home. It's a win. We did great. But the person can't, I mean, you can look at gross things like, oh, I can't play golf anymore or something like that, but they may not be able to even just take care of themselves and need help. And the help to provide to them is difficult to come by. I mean, there are people like neuropsychologists that can help with some of the neurological, neuropsych issues that people have. But that's a whole area of research that really needs a lot more work because just living isn't really the end game. It's living and being able to do all the things you like to do and having a normal life and getting people that with any kind of critical illness is not as easy as it would seem.
I would like to comment too that the access both to these treatments and to that kind of therapy that you need to recover depends very much on who you are and where you are. And that is another big ethical question for society to figure out. If we're going to do this, and it's very expensive, as you made it clear with the number of highly skilled people involved dealing with one patient in the extreme situation, how do we deliver that equitably across society is a big question. Adding questions from the audience. I see people pointing, but I'm not sure who they're pointing to. Oh, I see the hand now. Okay.
Thanks. Howard Gardner here from Cambridge. Appreciated this wide-ranging set of presentations. The word ethics was touched upon, but I haven't heard much about religion or theology. And I'm wondering to what extent, as these kind of interventions become more widespread and more successful, what do people in different societies with different religious cosmologies think about these things, weigh in? Are there people or groups that object to this, or is this seen as simply an extension of medicine, which I don't think any group would be against? Thank you.
I don't think any of us is an expert on that. Well, maybe some of you are, but there's a rich literature about attitudes toward heart transplants and how different patients in different religious groups, different socioeconomic groups, different cultural, what used to be called racial groups, how they respond. And I'm sure there is a literature out there concerning these procedures, but I don't know what that literature is showing yet, if anything.
Yeah.
I can just comment on doing resuscitation research. As I mentioned, you obviously can't get consent from people when they're trying to die, but we do this community consultation. We go out into the community and talk to people, and we as researchers need to go to the people that are at risk of becoming subjects in our studies. So that could be fairly easy if we're talking about people having car accidents, because anybody who drives a car, or even if you're not the one driving, you could be involved in a car accident. So people sometimes go to community events or go to church events or other things and talk to people. It's a little more difficult with penetrating trauma. That's a very more confined demographic. But we did that. But then I can tell you, having done that in 2 different cities, it can be very different in terms of the responses you get. So some of that is related to previous history with the medical system. Some of it's related to religious beliefs. Some of it's related to the trust in not the medical system in general, but that specific medical system. So all that plays into people's views of it, and it's important for us as researchers or people trying to push this out there to know what people think about it, get their input, get their thoughts about it, and not just force it upon them.
I didn't have enough time, but I failed to mention that DARPA does have an LC component, so that's ethical, legal, and societal implications, and all the programs we could do. Some things within BTO, the Biotechnology Office, is pretty apparent when you're dealing with synthetic biology or human-machine autonomy. But some other programs that you might not think that has an ethical component actually does, right? When you're dealing with maybe collective intelligence or some of the other areas that we touch in the information office. And so we actually have a LC Scholar, dedicated resources within the agency, and we don't wait till public perceptions or complaints or transitions, issues for unintended or intended consequences at the end of the program. We actually start beforehand and have workshops to talk through, to bring theologists. So in fact, two folks on not this Biostasis program, but some of my other programs that are theologists and philosophers. They actually advise us on, what are the potential pathways that we go down here in terms of ethics and society.
Yes, here in the front row.
Samuel Zeki, University College London. Is there any account at all of what happens to these, and do these patients have any thought processes which they relate after biostasis or after hypothermia or after anesthesia? And are these accounts consistent?
You mean of their experience during?
Yes.
During, yes.
I've had many patients tell me as to their experiences while they're either recovering from general anesthesia or after they've survived a cardiac arrest. One young man specifically, he was on life support, and we had to lighten his sedation in order to make sure he was neurologically okay. So we just asked very basic questions. Can you squeeze my hand, wheel your toes, shake your head, yes or no? And then we would intentionally resedate him. This was while he was on an ECMO circuit. And one of the things that he found disturbing, he's now, breathing tube is out, he's talking, he's discussing things with his wife, and what he relayed the experience of having recollections, and he wanted to know if that really happened. Is that for real? And so I had to explain to him, yes, we did this and this is why. So he thought he was like kind of losing his mind, and he was, very disturbed by it. But yes, that was real, and he was comforted by just the information that, yes, this is real, this is what happened. And also, too, his wife, what she described is that she witnessed all of this, and it was very traumatizing to her to witness him being on just death's door, really. And he slept through all of it. So he and her had a different perception of that experience. So I counseled them that, this is probably a good thing to recognize. Your experiences of this life-saving issue were completely different and traumatizing to both of you for different reasons. So I encourage them to talk to our psychiatry department, and let's start a relationship where you can talk about it.
So what I want to understand, they are unconscious during the process, but they are conscious in the sense that there's content to their unconscious state. Is that right? Would that be a correct description?
How awake or aware someone is, and I don't have them necessarily hooked up to an EEG monitor, so I don't know that level of awareness, but I know that they're unresponsive. That's what I can say. So I do think that there are probably memories that are being formed from that experience that we probably don't have a good description of. But he clearly, because we were lightening his anesthetic, that he clearly had clear memories, and they were disturbing to him, and he was hearing sounds or machines, and so.
You're in a very strange environment. We're coming to the end of our time, so if there's one last question somebody was really urging to ask, can I see a hand? Who get Bob? Last question, Bob.
It's another of my, is there anything to be learned from questions, and that is, what about IVF? where you have frozen embryos. Isn't that, is that in some sense an instance of a successful biostasis? And is there anything to be learned about that relative to what you folks are engaged in?
Yeah, so my specialty is history and philosophy of developmental biology, and that's actually an issue we've been arguing about with my graduate students. And how would we know? Does that count as biostasis? The answer is that since we don't really understand the range and umbrella of what comes under biostasis, we can call it that if we want to, but really what's important is define our terms as clearly as possible and be as clear as possible what we actually mean.
Yeah, thank you. And thank you everybody for your wonderful questions.
What? You want us to stand up?